bells are ringing. Welcome, everybody. My name is Teun Dekker, and I will be the pro-rector today, representing the rector under whose authority we will hopefully confer the doctorate in a little bit more than an hour's time. I'm very happy that you are all here, especially to the audience that has come to support the candidate in this exciting ritual, because it is a ritual, and even though it looks like an interrogation and there will be formulas and critical questions, in the end we should never forget that this is a celebration of science. These little booklets, they seem so small and insignificant, but they take an awful lot of time and effort and skill and sheer determination to will into existence. And the fact that it is now here is something you should be very proud of, and that we should celebrate here today. And with that, I hereby open this academic ceremony in which Yanyun Zhang will defend the academic thesis entitled Microbial Degradation of Structurally Diverse Plants-Derived Components, Insights from the In Vitro Colon System, TIM 2. Dear candidate, may I invite you to present a summary of your study and the conclusions of your thesis.
Thank you, dear Pro-Rector, for your nice words. Dear Assessment Committee, dear colleagues, friends, and family, thank you for attending. In the following 15 minutes, I would like to give an insight of my research, microbial degradation of structurally diverse plant-derived compounds, insights from the in vitro colon system, TIM 2. I would like to start from a brief introduction of the background. The gut microbiota is a diverse, dynamic environment in our gut, comprising trillions of microorganisms that form a mutually beneficial relationship with the host. The following two terms will be mentioned very frequently, so I would like to introduce them here. They are microbial diversity and microbial community structure. The microbial diversity means how many different kinds of the microbes are present in the gut, so literally diversity. And the microbial community structure means which types of the microbes are present, so it's more specific. The changes in the microbial diversity and the overall community structure can affect the gut microbiota, and consequently it will infect the human body. A lot of research already found that the gut microbiota are not only related to the digestive health, but also like the mental health and even the cancer, which means it's really important to our health. While the gut microbiota resembles a unique fingerprint for each individual, the composition can be influenced by many factors, and diet is a very important factor shaping this complex community. As the saying goes, you are what you eat. Particularly, dietary fiber plays a key role in shaping and sustaining a healthy gut microbial community. Of course, there are other ingredients in the diet that can also influence the gut microbiota, but because of the time limitation and also the importance of the dietary fiber, today I'm going to only describe dietary fiber here. So basically, when we intake diet, we absorb nutrients from diet. However, our body cannot digest dietary fibers because we are lacking of some important enzymes. But the gut microbiota can produce these enzymes, which allow them to eat dietary fibers. We call it digestion or fermentation. After the fermentation, the microbiota composition will be changed and some metabolites will be produced, especially the short-chain fatty acids, which can influence the host health. So what is dietary fiber? It is defined as carbohydrates containing more than 3 sugar units that link by the glycosidic bonds, which can be utilized by gut microbiota. The structure of dietary fibers can vary a lot depending on many factors like the size of the fiber or the type of sugar units they contain. For example, inulin-resistant starch and pectins, they are the common dietary fibers, but they have very different structures. So the icon here with different color represent the different types of the sugar unit. So a dietary fiber containing more colorful icons means that the dietary fiber containing more different types of the sugar units. And when a dietary fiber containing more sugar units, it will have a bigger size. And a lot of research already found that the size of dietary fiber and the type of sugar units can influence the fermentability of dietary fiber. However, in the past, most studies about dietary fiber often didn't go into too much details about the structure. And this study usually focus on one single type of the fiber. So we are missing a clear comparison of the different dietary fiber tests side by side under same conditions. Therefore, the aim of our research was to investigate the effects of structurally diverse dietary fibers on the fermentation rate, the microbial metabolites produced, and the change in the microbiota composition using a standardized approach. In chapter 2 and 3, we focus on 8 soluble dietary fibers that from different food sources, which were obtained from the industry. And in chapter 4 and 5, we focus on 3 hemicellulose fractions that isolated from the sugarcane bagasse, the byproduct. So here's the method we used in the complete research. So first we applied different analyses to determine the structural features of dietary fibers. We used HPSEC to determine the size of the fiber, use GCMS to determine the which type of sugar units they have, and we use FTIR and NMR to confirm the results from GCMS and get more information about the structure. After we determine the structure of dietary fibers, we would like to know how they influence the gut microbiota, so we use a standardized TIM2 fermentation for investigating the microbiome effects of this dietary fiber. TIM2 is a computer-controlled system that can mimic the nudge in testing. For the fermentation, we divide it into 2 parts. So first we apply them in a 72 hours continuous fermentation in chapter 2 and 4 to simulate the intake of dietary fiber on a day-to-day basis. After that, we would like to know how this dietary fiber to be degraded and is there any key degrader for this dietary fiber. So we applied a 24-hour single-dose fermentation in chapter 3 and 5. And here's our result. So in chapter 2, we test 8 different dietary fibers, include 1 resistant starch, inulin, arabidoglycan, 4 different pectins, and 1 pectic derivative. And we found all of them, they have different size and the sugar type. Then about the microbiome effects, we found they have very distinct effects on the gut microbiota. For example, the SRS and the PS3, they had really similar changes in the microbial diversity and microbial community structure, but they have very distinct size and sugar type. In the other hand, SRS and inulin, they are both small fiber with only 1 sugar type, but they have distinct microbial diversity and the overall community structure. So now it seems like the microbial effects of dietary fiber are not really rely on the size and sugar type. As for the metabolite production, we observed that all dietary fibers can improve the total short-chain fatty acids production, with pectin being the most pronounced in the short-chain fatty acid production and acetate production. So from chapter 2, we found that the microbiome effects of dietary fibers cannot be only explained by the size and the sugar type of the dietary fiber. Then we would like to know how these fibers to be degraded. So we detect their degradation rates. And we found that different dietary fibers, they have different degradation rates. And it also seems not really related to the size and the sugar type. For example, like the similar dietary fiber inulin and SRS, they have very distinct speed on the degradation. And HG, compared to SRS, the structure is different, but they have very similar degradation rates. As for the microbiome effects, we found very limited effects of this dietary fiber on microbial diversity after 24 hours fermentation, and we didn't see very clear effects on the microbial community structure and bacterial genome. But this dietary fiber still can improve the short-chain fatty acid production, and pectin still resulted in the highest total short-chain fatty acid production. So from chapter 3, we found that each dietary fiber inhibited unique degradation rates, which cannot be explained by size and sugar type. And unfortunately, we cannot identify the key degraders because of the limited effects. So now we move our focus from 8 different dietary fibers from different food source to 3 hemicellulose that isolated from the same food source, the sugarcane. So first we determined the structure of the dietary fibers, and we found this, even though they come from the same food source, they have different structural features. The F1 is the biggest one, which mainly comprising of xylose, and F2 and F3, they are both smaller dietary fiber, and they have very similar composition in glucose. F1 causes a decrease in the microbial diversity, and F2 and F3 cause they can sustain the microbial diversity, and they resulted in a very similar change in the community structure. About the metabolites production, all of them can improve the total short-chain fatty acids production, but F1 mainly induce the butyrate production, while F2 and F3 induce the most propionate production. So from chapter 4, we found that even though the dietary fiber from the same food source, they still have different structure features and microbiome effects. And interesting now, it seems like the size and the sugar types, they are matter. But are they still matter for the degradation rates? We also checked how this dietary fiber to be degraded, and we found that these 3 hemicellulose had very different degradation rates. And now it seems like the degradation rates of this dietary fiber are not really related to the size and the sugar type, because as we mentioned, F2 and F3, they are more similar in the size and the sugar type, but they have very distinct degradation rates. And as for the microbial effects, it's a similar result we observed from chapter 3, that after 24 hours fermentation, this dietary fiber had very limited effects on the microbial diversity and the overall community structure. But they still can induce the short-chain fatty acid production. So F1 still induced the highest butyrate production, and F2 induced the highest propionate production. So from chapter 5, we learned that smallest hemicellulose is more rapidly degraded, and still we cannot identify the key degraders of these dietary fibers. So here's the general summary of this thesis. No matter 8 diverse dietary fiber from different food source or 3 hemicellulose fractions from the same food source, they all have different structure features and distinct in the microbiota composition, metabolites production, and degradation profiles. For the 8 dietary fibers, the size and sugar type are obviously not sufficient to predict the microbiome effects. But for the 3 hemicellulose fractions, the size and sugar type, they seem matter because they influence the microbiome effects, but do not tell everything. So the conclusion of this research is, first, we confirm the potential effects of dietary fibers on changing gut microbiome. Then the hemicellulose from the sugarcane bagasse have the potential health benefits. The structurally diverse dietary fiber influence the gut microbiome differently, and the net is more studies are still needed to explore the structure-function relationship beyond the size and the sugar type. This is the end of my presentation. Thank you for your attention. Now let's give the word back to the prorector.
Thank you so much, dear candidate, for that fascinating presentation. Next time I will make jam, I will make sure to add extra pectin. Good for you. The opposition will now be opened by Professor Ellen Black, who is Professor of Human Biology at the University of Maastricht.
Thank you, Mr. Prorector. Dear candidate, of course, at first I would like to congratulate you on this very nice thesis. Describing fermentation characteristics, microbial degradation of different plant-derived fibers. And of course that provides very useful information on the structure-function relationships which can benefit in the end health. But of course I'm sitting here to ask you some questions, and I would like to start off with the different models you used. You used the TIM2 model, but you had several differential protocols. Short-term, 24 hours, long-term, 72 hours. And you make a little bit of comparison between both, also in your discussion and presenting several figures. I just wanted to know if I would like to perform a human study and execute a screening in before with many different fibers, and I would like to look, for instance, at short-chain fatty acid production. What model would you advise me? So what can be best translated into human conditions?
Highly esteemed opponent, thank you for your question. If I need to apply the results in the human body, I would recommend for 72 hours fermentation. Because first, the setting of 72 hours fermentation was based on the daily intake. So it's more fit in the realities. And from our results, we can see that the short-chain fatty acids production, so the ratio of each short-chain fatty acids production, they are matched for the production that from previous study that people observed from the human. So it proves that our research reflects more conditions in the human body. And also for the 24 hours fermentation experiment, the purpose we set it up is to track how dietary fiber to be degraded. So it's not, so we only give them one shot at the moment, and it's not realistic because, like in your gut column, there will not be only one food coming and the transit stopped. When you're eating, the food will keep going in your column. So for 24 hours, it's not applicable for the clinical trials.
Okay, and just have a few follow-up questions. At first, you indicate 72 hours continuous feeding does reflect human conditions, but we eat most of the time in 3 meals a day, and we also sleep during the night. Does that compare to you? Each 2 hours continuous administration? Or?
So even we eat 3 times, but yeah, we intake food 3 times. But when we digest the food, it keeps transiting and it didn't stop. So our system in 72 hours just mimics the transiting and also like squeezing all the times.
Okay, just a little bit on the short-term model, because you indicate 1 shot.
Yes.
And then, of course, the dose is also of importance. You would like perhaps to have a dose that really reflects daily intake. So why did you choose for a dose of 1.5 gram?
So the purpose we set the 24 hours models, first we want to track how dietary fiber to be degraded, and we also want to identify the key degraders. So we would like to create a competitive environment for the key degraders. Then we decide...
just, sorry to interrupt, but why not use, for instance, a higher dose of 7.5 grams?
Well, that was, we got references from previous study. Then we decided just to have a try to see if it also work on in our cases. But from our results, unfortunately, we found that the 1.5 dose is too low. So even though we still tracked how dietary fiber to be degraded, but we cannot find the key degraders. That's a pity from our research.
When it's a higher dose, or your dose, you mean?
With my dose, because my dose is too low.
I was just wondering, because you indicate also cross-feeding may not occur that much in the short term. Would it make a difference if you differentiate between proximal and distal fermentation, for instance, by increasing pH?
Actually, it can be, I think, because like for some dietary fibers, they are like slow fermented. So maybe in the real condition, when we eat them, they are not be degraded in the proximal colon, and when they transit to the distal colon, they probably have different effects. And not only the cross-feeding, but the different species that located in the distal colon, maybe they are more the key degraders for specific dietary fibers.
Okay, and I would like to end with the last question. I will have a question on proposition 8, and I will read it then myself in view of time. And dietary guidelines should shift from simply urging more dietary fiber to emphasizing variety of dietary fiber types, since there may be different effects on microbiome. When you look at, for instance, dietary guidelines, they're not dealing with supplements. There's an increase in dietary fibers through various food-based approaches and products like whole grains, legumes, vegetable, fruits, and nuts. So that's a large diversity. My question is, should we go beyond that, or would you say when you eat a sufficient amount of fiber with food-based within different food products, that is sufficient?
Well, I always recommend people to eat the natural food, but maybe that's not really sufficient when you really have some need. Because, like, for example, the food guideline recommend people to intake 20 to 25 grams of dietary fibers, but dietary fibers first containing soluble dietary fibers and insoluble dietary fibers.
But the food-based guidelines also advise you to take a wide variety of foods containing both insoluble and soluble fibers.
But it's about the containing of the soluble dietary fibers. Like, for example, if you just eat sugar beet, you want to intake soluble dietary fibers, that will be 1 to 1.5 gram per 100 gram, the sugar beet. So if you want to just got enough soluble dietary fiber from the raw one, then that will be more than 2 and 3 kilos. You have to eat that per day. So that's not realistic.
Okay, I think it would be good to discuss that a little bit further afterwards. I'm happy with the answers and give the word back to the provost.
Thank you so much. The opposition will now be continued by Professor Hauke Smits, Professor of Complex Microbial Ecosystems, all the way from Wageningen University. Thank you so much for coming.
Thank you, Mr. Prorector. Dear candidate, first of all, also I would like to congratulate you with the completion of your thesis, which I read with a lot of interest. I learned a lot about fibers, how to measure them, and the broad variety of these compounds. And so I was very much intrigued by the content, but also the outside that we also see on the slide there on the screen. And I always try to understand whether the cover is related to the content, and in this case I think that's obvious. So we see different types of fiber-containing foods. At first I have to admit I missed the microbes, but then if you take a magnifying glass, I think they're these little dots, right? So when you now think about the content or the aim of your thesis, which is dealing with the interaction between fibers and the gut microbiota, which of these two components do you think is more important for health? If I look at the picture, I could get the impression food is important, microbes don't really matter because they're so small. So how do you see that, also based on your own research?
Thank you, highly esteemed opponent, for the question, and thank you for noticing the dots, because it's really tiny, but I did that on purpose. So I wouldn't say which one is more important, because the relationship of them, like, they can not miss each other. The microbiota is like stay with us since we were born till we die. It's kind of like the in vitro organ of ourselves, and it can, it brings benefits to us, and sometimes it brings the disadvantage to us. And for dietary fibers, it seems like not related to us, but they are the energy for the gut microbiota. So, when it just supports the gut microbiota. So if gut microbiota without dietary fiber, of course, the gut microbiota can also digest other things like the proteins, but they mainly produce BCFS, which will influence, which will cause disease. So I wouldn't say which one is more important, because they are inseparable.
Right. So then maybe we can go to some of the studies that you did. So you looked at the effect of different dietary fibers and how they potentially modulate gut microbiome composition and function through the metabolites. And you and many others also do that, said that dietary fibers potentially benefit gut health or human health, and they do that through modulation of the gut microbiome. So how do I have to see that? Is change always good? Or how does that modulation need to look like?
Well, so when we say the dietary fiber can modulate gut microbiotas, for some of them they can keep a high microbial diversity, which is also good for the diet of the human, and then they help for producing short-chain fatty acids. And short-chain fatty acids, for each of them, they benefit for like the entire gut environment, or the liver, or the colon cells. Even though, like in our research, we didn't have the really direct experiments on how these things can influence the humans. But other research already found like the short-chain fatty acids has really good effect on humans.
Imagine I'm eating. Right, I have a varied diet with lots of vegetables and fruits and nuts, as we just heard. Would I actually want these fibers or these fruits to change my gut microbiome, or?
Well,
so if you are healthy, is change good, or would you actually expect it to change?
So if you are healthy, we expect that the dietary fiber can help to induce the short-chain fatty acids production, and to decrease the branched-chain fatty acids, so you will not get like the inflammatories, and then like prevent you from some disease. So that's why we recommend the healthy people to intake the food. But for like people that unhealth, like if they are very sensitive for the gas production, and our research, like fibers induce a lot of short-chain fatty acids, that would be not good for them.
Right. So maybe is there time for a small question? Let me get back to the dots on the slide there, because I sort of assume that those represent the bacteria in the gut, and there's hundreds of trillions of bacterial cells. But in your introduction, you also mentioned other types of microbes. You mentioned archaea and fungi, protozoa, and actually also viruses. I guess those are not really shown. You also didn't study them, but should we actually do that? And if so, why do you think we should study viruses, for example?
Well, now we call it gut microbiota, but the microbial community is very complex. And because we are focused on the dietary fiber, which is the direct energy source of the gut microbiota, so that's the reason why we only focus on gut microbiota. But of course, and also because gut microbiota is the most abundant member in this community. But of course, other members like the fungi and the virus, they will also, of course, influence the gut health. Our not our previous study, but previous study already found that the fungi also will influence the gut microbiota, and sometimes they have the interaction with gut microbiota. But they are not the direct consumers of dietary fiber.
And I want to give the word back to you, Mr. Prorector.
Thank you. The opposition will be continued by Professor Paul de Vos, freshly minted Professor of Sustainable Food and Immunology at Maastricht University.
Thank you so much. First of all, I would like to congratulate you with this beautiful thesis. I learned a lot actually, and involved in my congrats also the supervisor team and the co-supervisor. Of course, I have a number of questions, and I would like to kick off, which I hope is a rather simple one. I know the TIM2 model was a vendor when you started, but could you elaborate on why you actually prefer the TIM2 system rather than other dynamic in vitro systems such as the chyme or batch fermentation? And what do you consider the main added value of the TIM2 model?
Thank you, highly esteemed opponent. And so I will first answer why we choose TIM2. So TIM2, as I mentioned, it's an in vitro model that can mimic the colon intestine. So it can really mimic the the majority condition of the colon, like the temperature, the transit speed, the pH, and the main point is it has a membrane that we can really detect the how the short-chain fatty acids be produced, which like others, like the chyme, they cannot really detect the metabolite production. So this is an advantage for TIM2. And also for applying the experiments on TIM2, it needs less time. For in my case, I have 72 hours experiments and 24 hours experiments. So for whole duration to apply one experiment, the maximum is 1 week. But for chyme, that will be 1 month. So it's also more efficient.
Thank you. Then I would like to change gears a bit. I noticed already in your lecture that you're quite enthusiastic about pectins. Me as well, I'm a little bit addicted to it. And you tested actually different pectins and pectin-derived substrates, and including the one that actually is RG1 rich, which is at the moment really very famous because Nutrients is bringing that on the market as a type of pectin that is immune stimulatory. I don't really know whether it's really the case, but that is a sidestep. Could you explain why pectin were particularly of interest in your model, and how you feel that variations in degree of methylation and molecular weight in your experiments led to different microbial responses?
Okay, so why we focus on the pectins? First, so pectin just present very commonly in fruit, and it's kind of like the most complex one. It containing the most different types of the sugar units, and generally the sugar size is also big, so we are interested in the pectins. And research says the structural diversity of the dietary fibers will influence their fermentability. Since we focus on the structure-function relationship, we think that the more complex dietary fiber will bring some interesting results to us. And this is the reason why we focus on pectins, and we chose 4 different pectins. And yes, we also observed these 4 different pectins resulted in very distinct results in the microbiome effects and also the metabolite production.
If I saw it well, they are different in methyl acidification and in molecular weight.
Yes.
Do you have any idea why that has led to different microbial responses?
Well, so I think we didn't detect, but I think it may related to the different glycosidic linkage they have, because basically the monosaccharide composition, so the monosaccharide types, they're similar, and they also have the GLC, and also they have different level of the DM. But due to the different contents of the monosaccharides, So they must have the different glycosidic linkage, and yeah, so different. So as we said, why gut microbiota can consume the dietary fiber, because they can produce the enzyme to break down the glycosidic linkage. Since they may have the different glycosidic linkage, they will be selectively utilized by different gut microbiota. So I think it is the main reason why it causes the different microbiome outcomes.
It'd be a suggestion that it actually is differences in cross-feeding between microbes.
That would be also.
So time for another small question of personal interest as well. So the TIM2 model is here at Maastricht University, but for example in Belgium they are quite fan of the SHI model. The SHI model is evolving at the moment. They are trying to adapt it as such that you can more adequately study host microbe interactions. So I know that some people, it's not published yet, but they are using also epithelial cells, that kind of stuff, and you did some suggestions in your thesis. What do you feel we should do in the future to change the TIM2 as such that we can also study really microbe host interactions?
So as we mentioned, so even though the TIM2 can mimic the condition, it still has the disadvantage as with that, like in linking the immune system interactions, it doesn't contain any cells. So from our research, we can only see the effects of our test products on gut microbiota. So if we want to see more about the microbe-host interactions, we can apply it for more cell experiments after or before we start the TIM2 experiments, because not only the end products like the short-chain fatty acids will have the interaction with the cells, but also dietary fiber itself also will influence the immune system. So I think that's something we have to do.
Sorry for interrupting, but I have a little time. But what do you feel about inserting, for example, a compartment where you have epithelial cells, or where you can add organoids, where you have more interaction between the intestinal lumen and the cells? Do you think that's possible, or?
Actually, we plan to do. So I used to have a plan to apply experiments that we extract the supernatant from the lumen, and we see try to see if the short-chain fatty acid left, if it will affect the cells or not. And so I think it is doable to try to see the products from after the fermentation, if it will have the interaction with the cells. Of course, there's more factors we need to consider, like if there will be contaminations or not, but we can also use other way, like for the stereo filters, and to see if it will have the effects on the cells.
Thank you. I'm happy with your answers. Give the word back to the director.
Thank you so much. The opposition will be continued now by Dr. Osvaldo Hernández, Doctor of Science in Food Technology and specifically Carbohydrate Analysis at the Institute of Food Science Research in Madrid.
Thank you so much, Professor. I would like to congratulate you for your dissertation thesis. I'm very happy to see that the carbohydrate structure is taking into account of many biological activities. I also would like to thank you for the invitation to your supervisors. It's really nice to be in a different place from time to time. So of course I crossed Europe to ask you questions. So the first question that I have is related to the chemistry, to the analytical chemistry of carbohydrates. I realized that you use different methodologies to analyze the monosaccharide composition. Why was that?
Thank you, esteemed opponent. I have to say I'm not the person who really analyzed the monosaccharide composition, but from the literatures, the GCMS is the most common one to detect the monosaccharide composition, and I think that's why we use this protocol. Our collaborators, they applied that for me.
Okay, that's true. Do you think that carbohydrate analysis is an easy task. I'm asking you this because right now we have different omics, proteomics, metabolomics that is very well developed. What do you think about carbohydrates in general? I mean, part of your thesis is structurally how the structure influence the biological activities. What do you think about the carbohydrate analysis in general? Is easy, not? Why not many people is doing that? What is your opinion in general?
Okay. It's a really tough question actually, because, but I think are you asking like why now people more focus on the
structural
analysis
in carbohydrate? Because, for example, if you read different publications, who is doing, for example, proteomics. It seems quite easy. It's not easy, but nobody talk about carbohydrate analysis so much. Actually, there are few groups in the world doing that. Do you think maybe it's because it's quite complicated?
Yes, actually, it's quite complicated. Like we did a lot. We use like very varied methodology to determine the structure, like the HPSEC, the GCMS, the FDR and MR, but we still didn't have the full picture of that dietary fibers. The structure seems really complex, and not a lot of people, they really go into to see like, oh, is this part of the structure will really influence this or not? Most people only see the overall, the general influence on their specific target. So most of people, they didn't really get into the details of the structure. But at least for the gut microbiota part, since we know the way that the gut microbiota consume dietary fiber, because they can break down the glycosidic linkage, which means the structure matters. So I think it would be, so if you want to analyze something, you have to understand why it can consume. So it's really important to know why the structure of that carbohydrates.
Yes, actually, that is correct what you just said. I mean, we need to understand the structure. Otherwise, you can get conclusions that are completely wrong. In fact, it's very interesting that in the first part of your thesis, you say, okay, monosaccharide compositions and molecular weight doesn't play a role. But then, when you change to hemicellulose, you see that these two factors play an important role. So, if you have the opportunity to redesign your thesis regarding to the analytical part, the analysis of the carbohydrate, so you have monosaccharide composition, molecular weight, what would you add in the analysis? What else do you think in the structure can play a role?
Okay, so for now, even though from our results, we cannot say that the monosaccharide composition and molecular weight, they are not important, because they only influence some dietary fibers, but not work for others. So maybe some deeper structure behind will affect, like some functional group or the glycosid linkage patterns. And yeah, so if I have chance to add more analysis in my thesis, I would definitely to detect the glycosidic linkage. And maybe I will use more methods to reduce the side chain of some pectins to see if those parts are really like working on the part.
Actually, some of your colleagues in the Wageningen University, they have very nice protocols that we are applying. Yes. Actually in Madrid. So yes, you have here people with a lot of expertise to do that. This is a correct answer. I mean, you need to find functional groups, but also the linkage play an extremely important role. So in modifying the microbiota in the colon, but either in other parts of the body that contains microbiota. And my last question is related to inulin. We know inulin for many years, and you have plenty of data regarding inulin. Did you find differences between the results of your microbiota profile with your system, with other results that you can find in the literature?
Yes, actually, because we know that inulin, they are the most famous butyrate producer. They can induce the abundance of Faecalibacterium. But in our research, we found it indeed induced the butyrate, but just a bit. And also, we didn't see very significant differences on the Faecalibacterium abundance. I think it might be because of the microbiota we used. So most of the studies, they used either the individual samples or the pooled samples, but with very limited number, like 5 or 10. But in our cases, we used 44 different donors. So our goal was to create a huge pool, with as much as the different species. Then we can have a general insight. But also, because we created a huge pool, we put 44 different ecosystems together, and that's not realistic, and maybe it decreased the function of inulin.
Correct, that can be one of the reasons. So this is my last question. I don't want to take more time.
Thank you so much. The opposition will now be continued by Dr. Freddy Troost, Associate Professor of Food Innovation and Health at Maastricht University.
Thank you so much, Pro-Rector. Dear candidate, congratulations also on my behalf. It is really nice after seeing you many years in the lab. It is in my hands. Congratulations. Also, I want to congratulate the team on my left side here. Having said that, I'm running to my questions. I come from physiology. I love humans. I want to know to understand what's going on in human beings. So with those eyes, I read your PhD thesis, and I try to learn from that. So because I'm new to this field, I have some questions on methodology, and then I will try to understand what your findings mean for physiology. It connects to previously asked questions. Professor Blaak already mentioned something about the duration of your experiments, 1 day, 3 days. But if we look in a human situation, an intervention would typically take at least 3 weeks before we reach stabilized homeostatic conditions in the gut. Microbiota changes over time, and if you feed something for a few weeks, that stabilizes. You do a 3-day intervention, and your response to Professor Blaak was that is because it resembles dietary intake. But it resembles dietary intake over a 3-day period. I checked the TIM systems. It should be possible to run a few weeks study and not only a 3-day study. In my opinion, from physiology perspective, it would make more sense to have a 3 weeks intervention. So why did you opt for the shorter duration of your experiments if the end goal is to compare results to human situation?
Thank you, esteemed opponent. It's really a nice question. Actually, at the beginning of the starting of the experiment, I also questioned myself and my supervisor why we select only 72 hours. And because of our goal, we would like to compare the differences between dietary fibers. So our goal wasn't to see how the dietary fiber will eventually influence how the gut microbiota do. So we just need an insight for that. And also because we just need to have an initial picture of how dietary fiber influence gut microbiota. From the previous study, we learned that the experiment, the fermentation in TIM 2 will have the same change after 72 hours experiments. So yes, we can do more experiments
So I think I will interrupt you because time is running out. So you consider your work to be more pilot work than final work?
Yes, because...
Okay. That is interesting, and that brings me directly to my other methodological question. In your previous discussion, you mentioned that you pull samples of many individuals. In several studies, you pull 44 individual fecal donations. Yes. In another study, 34. Well, maybe a short question, why one or the other, but that's detail. Why pull so many people? Because I understand from the people from microbiology that this individual difference between individuals in microbial composition is of critical importance. So if you pull samples, you get rid of the individual profiles, the fingerprints, so to say, in microbial composition. So would you not lose a lot of information by pooling all those samples, and would you not be more happy in your research if you investigate the fecal donation of one individual? So why pooling?
So back to our goal, we would like to to have a general picture of like how this dietary fiber to influence gut microbiota, and we would like to know which is the key degraders of this dietary fiber. So because of the, like for individuals, of course, because of the different diet habit and also the lifestyles, they will have different gut microbiota. So maybe like they don't have the bacteria themselves that can degrade that dietary fibers. So, but we want to have a full picture to see how these dietary fibers respond. That's the reason why we created a huge pool, including 44 different people, because we want to have a general insight.
But then you put everything together and you lose the variation that you need for proper diverse functioning of your intestine, right?
That also, after we test, we found that's a disadvantage. And in fact, if we have the chance to keep doing this research, the next step, one of the next steps will be we apply on the individuals to see how the dietary fiber influence the individuals, because also like the huge economic system, you will have
Thank you so much. I see that I'm running out. Short final question. While looking through all this microbiota data, I did not see really the changes in like percentage or fold changes in microbial composition. Why did you not report that? Because that would be understandable for someone like me who does not like relative abundances, because I can hardly interpret it. So why not express changes in fold changes? Please do give a short answer.
I have to say, I'm not the person who applied the bioinformatic information. So when our colleague did that to us, we thought that we can understand it, and we didn't think about the layman people when they read about it, if they cannot understand the results a lot. So that's the main reason why we chose to represent the results.
Thank you. I'm looking at the prorector. I think time's out, right? I give the word back. Thank you so much.
Thank you. The opposition will now be continued by Dr. Niels van Best, Assistant Professor of Host Microbe Interaction and Microbial Ecology at Maastricht University.
Thank you very much. Dear candidate, first of all, congratulations with your wonderful thesis, and I also would like to extend these congratulations to the supervision team. I think you did an excellent job here. And I also read with pleasure your thesis and learned also a lot about the dietary compounds and fibers structure-wise. But it may not surprise you that I also have some questions, in particular related to the microbial analysis. And for that, I would actually like to go to page 109, figure 2a. So maybe you can start explaining what we see here. And also for the audience, so it looks like a square with light-colored confetti with stripes through it. So maybe you can start explaining this.
Thank you, esteemed colleague. So you want me to describe the figure 2 from pilot A to pilot F, or just
E and F, or E or F, yeah.
E or F. Okay, so if you have the book on hand, and we can see the how do I describe the shape with different colors, which represent different dietary fibers. For example, the gray represents the control, the green one represents the F1, the purple is F2, and the orange one is F3. And we can see that at time point 0, at time point 0, at the left part, everyone starts together, which means they all have the same, like similar community structure. And after we feed them with different dietary fibers, the dietary fiber influence them differently by time point 24, 48, and 72. When you look at more to right side, and you will see that the lines go to the different directions, which means
so if I summarize this, you see microbial changes in community structure.
Indeed.
And it looks like different branches all originating from one root. So also explaining that they all have similar baseline compositions, right?
Indeed.
Now, if you go back to your chapter 2, actually, we see there with the similar workflow something different, and then you see differences in baseline composition, both between the conditions, but also within the conditions, between the duplicates. So can you maybe explain how this could have happened, or why this is?
Okay, so let me explain first. So the different panels, they are uncomparable. So the analysis only happened within the panels. But yes, within the panels, for those two conditions, we still see some difference. So which means that maybe they don't have the same baseline. And we consider there's two reasons. First, we use 44 different donors, so 44 different ecosystems. And even though we connect them together and we incubate them together, but during the adaptation period, if you read that, that would be like 16 hours, and with 2 hours starvation time, it's a known duration. And these 44 different economy systems may adapt differently, which will cause these differences. And also because of the known durations and in the feeding, maybe something wrong with the system itself. We don't know, but we considered it maybe happened, like the transit stopping a bit, but it's not the main question.
Okay, so you see some differences in baseline there.
Yes.
And what does that in the end mean for the validity of the results, especially then in chapter 2 and perhaps chapter 3?
So even though they have the different baseline, but we still consider the results are reliable, even because the variety is not that large, and for the duplicates, they have the similar trends in the community structure change.
Okay, so in the end you see some changes in the general community structure.
Indeed.
You also look at taxonomic changes.
Indeed.
And one of the things that interested me most was my favorite bug, Faecalibacterium. So if I read correctly, this was stimulated by PS5, so maybe you can explain what I then need to eat, for instance. So where is this originating from?
So for PS5, sorry, I have to check the books. So it's pectin, and so the PS5 is from Lyme.
Lyme, okay.
Yes.
So, and if I read correctly, so you have, of course, short-term experiments and long-term experiments. Now I'm referring to chapter 2 and 3. But the Faecalibacterium was boosted within 2 hours in the short-term experiment, whereas it only increased in abundance after 2 hours in your long-term experiment. Now, I'm wondering, do I need to eat Lyme for 3 days? Or is like one mojito enough for me?
I cannot give the answer because even though we have like the insights from like 24 hours experiments or 72 hours experiments, but we applied them in the general pools. We don't know how it will affect in the individual. And also because of the cross-feeding, maybe other bacteria helped for the boosting. And I'm not sure whether in an individual it will also containing the Faecalibacterium and also the helper for degrading. So for now, I cannot recommend to like eating just like boosting just one time or continuous that. But just think about it. If we see that in the continuous feeding, it has the effect, and it's not bad to intake Lyme all the time, right?
No, especially not with this hot weather. So, and then continuing on chapter 2, so you test these significant differential shifts in relative abundance using Linda. I think that's a nice name, could be one of your paronyms. But however, I was a little lost when you stated that this was applied on 10 genera with the largest absolute absolute difference between T0 and T722. So can you please help me in clarifying this? So what are these steps?
Okay, so in chapter 2, so for the heat map, we just present like the top 20 abundant bacteria in the whole.
Yanyun Zhang, the time appointed for defending your thesis has now passed. The degree committee would now discuss the quality of your thesis and your defense. And so I request that you and your company leave this room. After the deliberation, the beadle will come to collect you to return here to this room.
Please. Put it on the ledge. It's okay. I'll pick it up for you later. This way, please. Audience, please.
Chef Alex.
Yeah.
Yanyun Zhang, the degree committee here present has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account your previous qualification, the degree committee here assembled has decided to grant you the degree of doctor. Professor Jonkers is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor now to take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Yanyun Zhang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affix it to the official seal of the university.
Please be seated.
Dear Yanyun, congratulations with this great achievement. We are very proud of you. You made very big progress since we kind of adopted you together with Khalidi more or less 1.5 years ago. And I think Khalidi is the one who knows you best, so it's my honor to ask her to give the laudatio.
Please stand.
This is my first time doing a laudation, so I hope I did it well. So, ladies and gentlemen here present, for everyone that is also joined online, and especially to her parents for following from her home country in China. It is a true joy and privilege to stand here today to celebrate Judy's achievement, but also to celebrate her as a kind, spontaneous, and creative person who became a very dear friend. Every day she still impresses me, and I still wonder where does so much spontaneity and creativity in her talks come from. It brings happiness and joy to all my days when I was together with you. And I say this from our team as well. For those who are not very close to Judy, I want to tell you a bit about her. When we started working together, it was still Corona time, so we had our first meetings online until I came to the Netherlands as a postdoc. After the lockdown, as soon as we met, she demonstrated herself to be an open and caring person. And we started to get along right away. We were ready and energetic to start our research in the gut microbiota field. At that time, I was a postdoc, as I said, following Judy in her PhD journey. She was always excited, very motivated, happy, and ready to face any challenge on her way. Actually, she still is. In our first times in the lab, she was clearly happy to be there, even though experiments with Team 2 could be quite unpleasant. She was always singing with a big smile on her face. Judy and I had quite a few stories together, and all of them I will remember with kindness and it always warms my heart. But two of them were memorable, and I would like to share them with you and bring a little bit of joy to this. First, she gave me the most precious present I could have here in the Netherlands, my cat. Do you remember that? Once going to work, she found a stray kitten in the road. When arriving in Villa Flora, she immediately came to talk to me, as she already knew I was very passionate about pets and animals. What could be a simple task became like participating in a 100-meter Olympics run, because we had to go to the place and run after the cat to be able to catch it. And that was quite an adventure. Not for my cat, unfortunately, because he was scared and afraid of me for 2 years. Yeah. Nowadays, he's very kind and also kind to her, right? He recognizes you and me, and I still think he remembers that day. The situation was not so fun when it happened, but nowadays we laugh a lot about it. Monica was also there. I wasn't sure if I should tell the story here, but yeah, let's do this. The situation described by her in her very creative way immediately after the episode was that we suffered a biological attack. Or maybe it was the poop revenge. So in another day in the lab, we were having problems with our Falcon tubes that were exploding. Also, Jessica and Son, I remember that. They were exploding in the freezers. And we came up with several hypotheses why they were exploding, and we wonder, yeah, let's go check it. So we went to the freezer room to organize these Falcon tubes that were exploding and also check if we could still do research because they were just exploding. And we start moving the tubes, and suddenly in a chain reaction, everything started exploding in the freezer room. And the whole building could listen to that. Everyone was out of the office to check on us because everyone could hear that. In summary, we spent the whole day cleaning the room together with Carlos and Monica. I won't give much details, but that was quite an event in Villa Flora. I think people still remember that. And we also, when we talk about it, we still remember. I believe we got famous after that day. But not everything was fun. A PhD journey by itself is already a difficult time, but I judge that in your case, you had a heavier burden. We had many difficult moments and moments of uncertainty when we didn't know if we could be able to continue or just finalize our research. We didn't know if our group would continue and even if we would be able to stay in the Netherlands. That causes us a lot of anxiety and sadness at some point, yet we kept going, and Judy was always strong, positive, and supportive towards our team. And to overcome these difficult times that lasted longer than we expected, we did stick together, rely on each other, and that paved a strong basis for us to continue. If that wasn't the case, I'm sure it would be very much more difficult. I also would like to thank Monica. She was a part of our strong team, so we are very good friends nowadays. And I believe that's why she is here as our paranymph. As if the current problems weren't enough, she also had one more extra heavy burden. She had familiar issues with her mom and had to deal with all of this together. Besides being supportive to us, she also went back to China for a moment to be supportive to her family. So as I said, when we are doing our research or we start a new journey, we see this as a straight line, but in her case, was full of hills, mountains, and difficulties. But also things started to get better when Professor Daisy and Professor John started to supervise you, right? And we were able to see what a supportive supervisor team is. And for that, I also need to acknowledge you, Professor Daisy and Professor John, and thank you to put a lot of effort in Judy's journey. What was not done in 2 years, you made it in this few times we've been together. Thank you so much for your help. I say in regard to Judy as well, because you embraced us and made this day possible. She also managed to work on her own. We were traveling together to do our research in Maastricht together with Judy, and life was very good and happy at those moments. We laughed a lot. We had so many nice moments together, either in the lab as work colleagues or outside of work as friends. We had numerous moments together that will be forever in our memories. Judy, you are a real winner. You did what some would consider almost impossible, possibly. The progress you made in writing, finalizing your thesis was impressive. I was amazed by how much effort you put to finalize the writing, how many nights of bad sleep, and how tired you got, and you kept going. Judy is a kind, generous with her time, and deeply caring. She supports those around her not because she has to, but because that's how she is. As her co-supervisor, I've had the privilege of seeing Judy grown in the doctor she has become today. But as her friend, I've been inspired by her empathy and her warmth and caring. Today is a celebration of all she has achieved. And I would like to thank you for being together with me, being my friend and my colleague at work. I will always remember you with a lot of kindness, and I hope we'll still be friends in the future. Thank you so much. Thank you.
Dear Dr. Zhang, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. A doctorate is forever, and it is partially about what you've done, but perhaps more about what you will do with it. I'm sure you will do great things with it, so we will follow your career with great interest. I hereby close this academic ceremony. I believe that we will retire to the stairs for a picture with the committee, and then we have the opportunity to congratulate the new doctor over there in the museum section of the wall. Thank you very much.
Dr. Zhang.